caffeine jolt. Okay, folks, let's get started. So a couple of things. One, the exams are available. And two, I did manage to get shut everybody up quickly. I did manage to get a grade distribution shown. The numbers are a little hard to see, but I think you can get an idea about where you stand. These are the sum of the scores of your two exams. So I was very, very impressed with the average again. Average was almost identical to the first exam. And as I probably have told you before, when I see a high average on an exam, I usually see it fall on the next exam, and you're one of the few classes I've ever had who did not do that. So that's really remarkable. I'm very impressed with that. And you guys also, I had one very high average last term as well. So either I'm getting softer in my old age, or you're getting better in your young age, and I like to think the latter is the case. So congratulations. That was a very nice performance. Some of you are struggling, and I recognize that, and if there's something I can do to help with that, of course, as always, let me know. But the class as a whole is doing quite well, and I'm very pleased with this. So that's where things are. The exams, as I said, are available for pickup in the Ag and Life Sciences Building, room 2011. And there's a key outside of my office, as always. So, okay. Shh. Thank you. All right. So I want to go further talking about transcription today, and we've got quite a bit of ground to cover. And so I want to get into that before, without any further delay, shall we say. Last time when I finished, I had just started talking about promoters. And promoters are special, important sequences because they define locations where to start transcription. It's important to note that the promoter is adjacent to the start site. So the promoter is always in the negative numbers, as I showed the numbering scheme last time. And I pointed out that when we look at, these are all prokaryotic systems here, but when we look in prokaryotic cells and we compare the sequences near the transcription start sites for hundreds of genes, we see what's called a consensus that arises of this sequence right here, TATAAT. And I talked about how the closer that any given these are called -10 sequences, the closest that any given -10 sequence was to that TATAAT, the more effective as a promoter it was. And by effective, I mean the more copies of messenger RNA that were made from it. So we talk about strength of a promoter. So a strong promoter is something from which a lot of RNA will be made. And a weak promoter is one from which only a small amount will be made. Now, as we will see, there are things that can modify or change how much is made, depending upon circumstances. But in general, what I've told you is what I've just told you is true. In addition to sequences at approximately -10, there's also in E. coli another common sequence that's seen at about -35. These are all approximate distances, and they are there. I'm not asking you to memorize these sequences. I certainly think you should know this is an AT-rich region. But other than knowing that there's a -35, I don't think memorizing the sequence really tells you much of anything. Now, why do we see specific sequences? We see specific sequences because proteins bind to specific sequences in DNA. All right? So we don't want random binding. We want to be able to control. And so proteins have evolved recognizing and binding to specific sequences. In the case of E. coli, that system is pretty simple. It involves an RNA polymerase. In some cases, it may involve one or two other proteins. In eukaryotic cells, it's much more complex, and there's an entire orchestra of proteins that may play roles in controlling transcription. And I'll say more about those later. All right. Well, one of the ways in which different promoters can be recognized with different efficiencies is told by the story that you see on the screen. It's not described very well there, so I hope to clarify that for you. I said that there were several proteins that made up RNA polymerase in E. coli, and I said we weren't going to pay much attention to what the function of most of those proteins were. One of those we're going to pay some attention to is called sigma. Okay? It's called the sigma factor. And the sigma factor associates with the RNA polymerase. And what it does is it helps to help RNA polymerase recognize a promoter. So sigma is the protein component, is the protein component of RNA polymerase that helps the polymerase to recognize those specific sequences. All right? Now, it turns out that E. coli have several sigma factors. And some sigma factors will recognize different promoters than others will. So now we start to see that as things change, if we change the sigma factors, the RNA polymerase may start recognizing and transcribing other genes compared to when that given sigma factor is not present. All right? So most of the time the sigma factor that's there likes the TATAAT box. It helps RNA bind to the TATAAT box, and everybody's happy. Okay? But there are circumstances that cells go through where they have to respond to environmental change. And that's something that cells have to do a lot of. The more able they are to respond to specific environmental changes, the more likely they are to survive. Well, one of those environmental changes that happens is known as heat shock. Heat shock. I talked a little bit about heat shock last term when I talked about folding of proteins. If we think about what happens when we heat up proteins, what happens, what did you learn from last term when we heat up proteins? Yeah, they denature, right? So we could imagine that if a cell gets a minor heat shock and it can't respond to that, it may die. So certainly if we get it too hot, if we boil everything, then the cell isn't going to be around to do things. But if the temperature goes up 3 or 4 degrees, all right, it would be useful for the cell to be able to respond to that because that's not a drastic change. Well, in E. coli, it turns out that they have a special sigma factor that is made in response to heat shock. So when heat shock occurs, a different sigma factor than normal is made by the cell. We're not going to go into the mechanism about how that's controlled because it's fairly complicated. But suffice it to say that when a cell gets heat shock, it makes a different sigma factor. Okay? Well, why is that significant? That different sigma factor interacts with RNA polymerase, and now, instead of taking RNA polymerase to all of the regular TATAAT boxes, it takes it to a different set of promoters and helps them to be transcribed. You see a couple of them on the screen. Here's a heat shock promoter. All right? Look at that. It doesn't look anything like a TATAAT box, but this sigma factor will recognize this. So let's think about, yeah.
What's the ends?
Ends mean any. Any nucleotides. It'd be GAT or GAU or C, right? All right. So whenever you see N in a nucleotide sequence, it means any of the 4 bases. All right. Now, what does this mean? Well, it means that if the cell is not experiencing heat shock, the regular sigma factor will not recognize this, and it's not making heat shock proteins. However, when heat shock occurs, sigma factor is made that recognizes this, and now there's a whole set of heat shock proteins that get made. And these heat shock proteins help the cell to survive the shock of heat. So I talked last term a little bit about chaperonins. Anybody remember what chaperonins are? What do?
Help protein folding.
They help proteins to fold. Chaperonins they have heat shock promoters. So you get heat shock, or not, the E. coli gets heat shock, it's going to unfold some of its proteins, but now what does it do? It starts making chaperonins to properly fold proteins. It's a very cool system. Now, heat shock is not something that's unique to E. coli. Virtually every cell on the face of the earth has a set of heat shock proteins that allow it to respond to minor heat changes that would otherwise kill it if it didn't fold its proteins properly. We have heat shock genes in our cells, for example, which is good because we get fever, right? Okay, so Different sigma factors allow different things. Here's one for nitrogen starvation. We can imagine that when the cell starts running out of nitrogen, it's a different set of conditions than it would otherwise have. And so when that happens, a sigma factor that recognizes this promoter to turn on the genes for making the response to nitrogen becomes available. Now, in E. coli, these systems are very simple. We say, well, there's one sigma factor here that's made under this condition. There's a different sigma factor that's made over here under this condition. In eukaryotic cells, as I said, there's an orchestra. There are hundreds of possible iterations of different proteins and combinations that can be made. And no, you're not going to memorize them all. But we will talk about them in general when we get to there. In E. coli, it's relatively simple. Okay. Well, last time I went through a sort of a verbal description of what you see on the screen, which is what's happening during the process of transcription. During transcription, RNA polymerase has to be copying strands, and you can see that process sort of happening here. I think this exaggerates the size a little bit, but suffice it to say, we can see the process occurring. This is a segment of DNA that is being transcribed, meaning that RNA is being made, and it's being made by this big yellow guy here, the RNA polymerase. You'll see that the DNA strands have been pulled apart. As I described, there, up here, is the coding strand, and down here, the strand being copied is the template strand. You'll notice that the RNA-DNA duplex does not extend for the entire length of the RNA. There's this floppy end that's hanging off of here. So the further that this RNA polymerase moves along, the more unpaired this guy gets here, and more floppy end hangs off, and the more this duplex has to reform. Now, if we think about what's happening here, ahead of the RNA polymerase, what's happening? Well, in essence, the strands are getting pulled apart, right? Because we have to move forward. If we're pulling apart strands, what's going to happen ahead of that bubble? Well, we're going to have strain. We're going to have a lot of twists put into there, right? So there is, in fact, a topoisomerase in front of, this is called the transcription bubble. We talk about replication fork for DNA. We talk about transcription bubble for making RNA. There is a topoisomerase ahead of the transcription bubble that alleviates that strain. Well, look what's happening behind there. We have to start rewinding everything, remaking the duplex. We're going to have the opposite phenomenon over here. Up here, we're going to have too many twists, and back here, we're not going to have enough. And yes, it takes a topoisomerase to fix that one as well. So on either side of a transcription bubble, we have topoisomerases that are adjusting the base pairs per turn appropriate so that the DNA will be relaxed. Okay. So this process occurs, if we look at transcription, and we'll talk about a similar thing with translation, we'll describe these as happening in three general phases: initiation, elongation, and termination. With E. coli, that initiation phase is studied considerably. And though I don't have a figure to show you about that, I will tell you a little bit about what happens with it. In fact, I've got a interesting protein here. Okay? Not protein, antibiotic. I'm sorry, my mind isn't working. It's called rifampicin. Okay? And rifampicin is a molecule that can bind to E. coli RNA polymerase. And it stops the E. coli RNA polymerase from being able to progress from the initiation to the elongation phase. There's actually a stepwise process in which these processes occur. The initiation phase occurs during about the first 10 nucleotides being synthesized in E. coli RNA. The first 10 nucleotides or so. During that time, the cell is setting up what's called an open complex. That is, the strands have been opened, and that open complex is being made during that initiation phase. The first few nucleotides are put down. Once those first few nucleotides get put down, the sigma factor goes bye-bye. So we got the first 10 nucleotides down. Once the sigma factor has gone bye-bye, we're in the elongation phase. Okay? What rifampicin does is it prevents E. coli from progressing correctly through that initiation phase. It never gets to elongation, and so it stops transcription in its tracks. Well, if you stop transcription in its tracks, you're going to, of course, kill cells, and that's why rifampicin is an antibiotic. Okay. Now, elongation is kind of like my bumper sticker the other day. Elongation proceeds, right? That's basically what happens. It just goes on until we enter the termination phase. Now, as I told you, what happens with the RNA polymerase in E. coli is it works in fits and starts. Up, back, up, back, up, back. And this is continuing throughout the elongation phase, moving like this. Okay? Up, back. Somebody asked me yesterday, when it moves backwards, does it chew out nucleotides? And the answer is yes, it does. Or yes, it can. Okay? All right. Well, we'll see that there are some considerations of the elongation phase as they relate to termination in a minute. But I want to turn our attention now to the termination phase, and then I'll come back and relate that to elongation for you. Okay? So termination. Termination, of course, is the process whereby we are stopping transcription, and termination is every bit as important as initiation. We don't want to copy the entire E. coli chromosome in a single RNA. Waste of energy, waste of time, and besides, we don't want to make all those proteins at the same time, in the same amounts. So having a specific initiation point and at least a relatively specific termination point is in the cell's interest. It's true whether it's a prokaryotic cell or a eukaryotic cell. Well, there's two general mechanisms I want to talk about for termination. They're called factor-dependent and factor-independent. The factor-independent is the first one I'll talk about, and it arises when an interesting structure, like you see on the screen, appears in the RNA. Now, I need to describe a little bit about this. Everything you've thought about with DNA base pairs so far has involved two different strands. One strand up here, base paired to another strand down here. However, it is entirely possible for base pairs to form within a given strand if the bases themselves are complementary to each other. You can see that this is an RNA in which what's called a stem loop, there's a stem, there's a loop, that is formed because these bases can actually form base pairs with each other. Okay? Now, this stem loop has a specific sequence in addition to having a specific structure. And what this guy does is, as the E. coli RNA polymerase is moving along, it's copying the DNA, it's making this RNA, it's making this RNA, it's making this RNA, and by the time it starts getting back down here, these bases start seeing each other, and they start forming a duplex. I like to think of this thing like the jack of your car. Okay? When you have a flat tire, you get the jack of your car out, and you jack up the car so that you can take the tire off, right? This guy provides lift just like a jack does. Remember, the RNA polymerase is sitting right here. It's making this thing, and the DNA strand it's copying is down here. All right? You got that pictured in your heads? This jack lifts up the butt end of the RNA polymerase. So where the RNA polymerase was sitting nice and conveniently on the DNA and making this thing, now its butt end has been lifted up. It no longer is sitting properly on the DNA. That starts the process whereby the RNA polymerase falls off. It falls off. And something that helps it to fall off is the fact that, look at the sequence of bases it just finished up with. A, U, 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 U. The weakest hydrogen bonds. This is the least stable duplex that we can have. The RNA itself is ready to fall off. Everything is ready to fall off, and you've terminated transcription. Now this is called factor independent because there's no protein that's necessary to make this happen. No protein is necessary to make this happen. The sequences themselves are built into the DNA, which of course is made into the RNA. And as a consequence, the RNA polymerase falls off. These fall off sequences happen at very specific places. That is, we have very specific points of termination when we have factor independent termination of transcription. Now some of these can be kind of elaborate and kind of cool. All right? So one of these that is out there, I'm going to show you what looks like a complicated figure, but it's actually pretty straightforward. All right? This is a gene for making a flavin, like FAD, only this one's called FMN, flavin mononucleotide. Don't worry about the name. Here's the RNA for this flavin. All right? If we look over here, this is a termination loop. This stem loop will act like a jack if it gets a chance to form. Okay? If it gets a chance to form, it will terminate transcription. Everybody understand that? So the question that we're asking on the screen is, under what conditions will this form, as we see on the left, or not form, as we see on the right? Well, on the left, it will form only when there's abundant FMN. Why? Because FMN will sit in this complicated RNA-looking structure and keep these sequences here from interacting with these sequences over here. As a consequence, these sequences over here can interact like that and make this stem loop. When we have plenty of FMN, what's going to happen? Termination is going to happen, and it's going to happen right here at this point. It's going to happen very early. The cell is not going to make any more RNA for making this protein that makes FMN, because there's plenty of FMN. On the other hand, if there's a shortage of FMN, there's nothing to stabilize this structure, and now those sequences that we saw base paired over here get tied up in this bigger structure. This guy will not terminate, at least not at this point, and the transcription of that gene will proceed. Now the cell will make the protein that's going to make FMN. Bingo! This is a beautiful, simple environmental response. When FMN is high, transcription terminates early, the protein is not made. When FMN is low, transcription proceeds, and the protein for making FMN is made. Now I'll stop at that point and take questions. Yes?
You say that loop causes the RNA to fall off.
Yep.
Right. But to me on the right with low FMN, that looks just like a huge loop.
Well, it does, but it doesn't have the same structure as this guy does. So one of the things you'll discover in RNA is that there's a lot of different possible things. Look at this guy right here that can form.
Yeah.
Because, for example, you can actually form a stable base pair with G. See that right there? So there's a lot more pairing possibilities. It doesn't make this specific structure.
So
It's the specificity of that structure that determines it. Very good question, a very common question. So what he's asking is, well, look at this guy over here. It looks like this jack ought to really be big. It really ought to kick it off of there very easily. But it's not that. It's the specificity of this structure that actually causes that termination to occur. Yes?
Does the FMN act as like an allosteric?
That's a good question. Does FMN act like an allosteric effector? Well, we think of allostery, of course, relating to catalysis in proteins. Technically, it's not an allosteric factor, but you're right. It is acting like one, because this is acting as a gauge of how much FMN is there. How much protein should I make so I make FMN or not make FMN? Yes, Jared?
Does the number of pairs in the RNA nucleotides slow the creation of protein?
Yeah, so it's a good question also. So his question is, this is called secondary structure, by the way. RNAs can have secondary structure just like proteins can have secondary structure. And it has some tertiary structure as well. This structure that's here can in fact affect how well translation goes on. So I'll save the answer to that question until I talk about translation. But yes, those do play roles in that. Yes, right here.
So when it's not terminated, that includes, when it's made completely, that includes the
That's correct. So this guy has perfect feedback mechanism. If I make plenty of protein, I'm going to have plenty of FMN, and it's going to shut down the making of more protein. I mean, it's a beautiful feedback system. And E. coli cells have a lot of different ones very much like this, where there's a very simple feedback to turn on or turn off the synthesis of a given protein. Yes, question?
So, like RNA, like copies from like 5 times 3 times.
Yep.
So won't it copy all that stuff before it gets to the nucleus?
It's going to copy all this before it gets to there. That's correct.
So what does all of that copy make the RNA do?
Okay, so our question is, well, it goes and makes all this stuff. What happens with this, basically? And the answer is it gets degraded. It doesn't do anything.
So the actual part it needs to make the protein.
The actual part needed to make the protein is further out here to the right. Now, you might say, well, that's a pretty good waste of energy, and it is a waste in a sense, all right? That there are nucleotides here, but making the proteins necessary for this are thousands of base pairs. So the cell invests a certain amount of energy in being able to use this as a barometer, which actually makes some sense to do. Does that answer your question? Yes, Luke.
Are there other molecules that will do the same thing, such as when FMN is high? So or is it only specific for FMN?
So this particular one is only specific for FMN. There are similar systems that E. coli uses to regulate amino acid synthesis. And it uses a very similar mechanism to this. Yeah.
So is this a second layer of transcriptional control? I mean, does it control initiation of transcription as well? Or
Yeah.
is that just constitutive and this is the only one?
Okay, so his question is, are there other levels of control of transcription in this particular system? The answer is no. So this is, and we'll see this happening frequently, that when cells have ways, needs to control certain genes, sometimes, depending on the gene, they may have a couple of different mechanisms in place. This one only has one, and this is controlling whether or not that initial sequence terminates or not. When we look at gene expression, which we'll talk about later in the term, meaning next week, when we talk about gene expression, we discover that not only do we control initiation of transcription or termination of transcription, we also control splicing, which we'll talk about in just a bit. We control whether protein synthesis starts efficiently. We control whether or not a protein is degraded. Okay? So there's a lot of different ways the cell can modulate how much of a given protein is there. And the number of different ways tell us, or remind us, you already know this, remind us that the level of any given protein is very critical for a cell to maintain. Very, very critical. We don't want to have too much or too little of any given protein. Okay. All right. Now, I've told you about the termination independent. Let's talk about the termination dependent, because it's kind of a cool one. The termination dependent system is not quite as precise. I'm sorry, not termination dependent. The factor dependent termination. Okay. The factor dependent mechanism of termination is not as precise as the factor independent. You'll see why that's the case in a second. Okay. The factor dependent involves a factor. And the factor is a protein called Rho. That little sign there is Rho. And Rho is a hexameric protein that looks like this. And you can see it's attached to this floppy tail that's hanging off of this transcription bubble. The way Rho works is it grabs a hold of the RNA and it starts shinnying up it. It literally shinnies up it. I always like to give the example of how many times in gym class in high school did you have to climb the rope? Right? First time I did it, I hated that thing. God dang, it was awful, right? Imagine climbing that rope, and instead of having the rope attached to something at the top, somebody's letting it out as you're trying to climb it. All right? It's a race between how fast you can climb and how fast someone's letting the rope out. That's what's happening here. This RNA is getting longer and longer, and Rho is shinnying up it. When Rho gets up here, when Rho catches up, and it will catch up eventually, what does it do? It jacks the butt end up of the RNA polymerase and everything falls apart. You've heard that story before. The factor catches up and allows that to happen. Okay? Well, how does it catch up? Well, obviously, Rho is going to have to work faster than transcription in order for that to happen. And it does. And sometimes it doesn't. What does that mean? Well, think about how that RNA polymerase is moving and slowing down and moving and slowing down, right? What's happening? When it slows down, Rho's getting a little bit of a chance to catch up. And I said there was another factor about how fast transcription occurs that I talked about last time. What's that other consideration that we have? What's that? Types of what? The base pairs, yeah. So what happens when the RNA polymerase hits a stretch of GCs? It's going to slow down. When we look at Rho-dependent transcription termination, we find that it more commonly occurs in regions that are GC-rich. Because this is where the Rho has had a chance to catch up with the polymerase and kick it off. Now, Rho termination sites are not precise. They're usually clustered, so we see very possible places, there's several possible places where they can end, and so we can get several possible RNAs that are made. But it's a numbers game. It's not going to make it too far. Once you go through a few of these, the likelihood that Rho is going to catch up is pretty good. Okay? So, factor-dependent, factor-independent allow the cell to control where transcription stops. Yes, sir?
So you have multiple possible endings to this because it's just sort of a general region where it's likely to end.
Mm-hmm.
Is that cleaned up later by different splicing?
So his question is that because you've got different possible termination sites, does the cell need to clean that up later? And the answer is it doesn't. It doesn't. So it's got no problem with the stuff that's hanging off there. Plus, this is happening in prokaryotic cells, and splicing occurs in eukaryotic cells. Yes, sir?
Is there an advantage to factor-dependent versus factor-dependent?
Yeah, that's a good question. Is there an advantage to one versus the other, and why do they have both? And I don't know the answer to that question. The advantage I would see is that the factor-independent, A, doesn't require protein, and B, is fairly precise. But if it misfunctions, right? So if that stem loop, for whatever reason, doesn't jack off the bad choice of words, doesn't doesn't jack up the RNA polymerase, what's going to happen? Then the transcription is going to continue, okay, and make a much longer transcript. So they probably 6 of one, half a dozen of the other. I'll make YouTube now, probably, I suppose, right? Okay. Any other questions? Okay. So much for termination. We terminated termination. Let's see, what was that? This shows, again, where you have various sites where you can have various links that are made. And the more of those sites that it encounters, the more likely termination is going to occur. Okay. Well, I want to jump down now and talk about tRNA and RNA in prokaryotes, because we're nearing the end of transcription in prokaryotes. In prokaryotes, there is an important thing to keep in mind. There's one RNA polymerase. That is one, there's hundreds of copies of it, but there's only one type of RNA polymerase. In eukaryotic cells, there are 3 types. Okay? So all of the RNAs that get made in a prokaryotic cell have to be made by the same RNA polymerase. These include the ribosomal RNAs, the messenger RNAs, and the tRNAs. If you don't know what those are, you should, so I won't go through those. The messenger RNAs are the ones we commonly think about, but of course, ribosomal RNAs and transfer RNAs are absolutely essential for cellular life. When we look at how the ribosomal RNAs and tRNAs are made in E. coli, they're generally made with one long transcript, and then the individual pieces get chopped out. There's a processing that occurs. So a very important component of transcription is processing. We'll see eukaryotes, there's a lot of processing that goes on. In prokaryotes, there's not a lot of processing. But this is one place where processing occurs, because in this system, we have to chop out, we have to cut up specifically the things that the cell is going to need. And this happens for ribosomal RNAs and tRNAs specifically. Okay. Now, there are some enzymes that help this to occur. And unfortunately, your book doesn't give us any figures for that, so I'll just briefly list them. One is called ribonuclease P. And it's responsible for chewing away and making a perfect 5' end of the tRNAs. Another one is ribonuclease 3, and it cuts out the 5S, the 16S, and the 23S ribosomal RNAs. Those are 3 different ribosomal RNAs that I'll talk about later. Okay? In addition, at the 3' end of tRNAs, there's a sequence that's added to all of them. This is a modification that occurs as well, and there's an enzyme that does that. I don't have the name of it here, but suffice it to say that tRNAs need to gain this CCA sequence at the 3' end. And the last thing that occurs for our purposes here is modification of bases. One of these is uridylate. You can see some of these modifications here. Here's a uridylate that's been modified to be a ribothiamidylate. For all those who tell me, and I get email from people who watch my YouTube videos and they say, there's no such thing, you shouldn't write DTTP because there's no such thing as ribothiamidine. Well, there is. There it is. And it's not just a fluke thing. In fact, when we look at transfer RNAs, especially in E. coli, they get chemically modified to make this. They get chemically modified to make this. All right? Transfer RNAs in both prokaryotic and in eukaryotic cells are significantly chemically modified. You see uridylate here. Virtually every base can be modified in a tRNA. Now, it's not completely understood in every case why some of those modifications are occurring. It may be that it helps to flag these guys as being separate from messenger RNAs, for example. It may be that it facilitates or prevents base pairing in some cases. But suffice it to say that transfer RNAs have a lot of chemical modification that occur to them. Ribosomal RNAs in E. coli will have some modification as well. Of the 3 types of RNA that we have in E. coli, the messenger RNAs are the least modified. Very, very little. That contrasts with eukaryotic cells where there's a lot, and we'll talk a lot about those. Okay? So in E. coli, it's fairly simple. tRNAs get chemically modified, ribosomal RNAs get a little chemically modified, and messenger RNAs don't get very chemically modified. No, you don't have to draw these structures or anything. Okay. That concludes what I want to say about prokaryotes. And I'd like to turn our attention now to say a few things about eukaryotes. Eukaryotic cells have a very different setup than prokaryotic cells. Very different. The playing field is very different for eukaryotic cells compared to prokaryotic cells. The playing field in a prokaryotic cell, remember there are no organelles, so everything is in this cytoplasmic soup that's over here. What you see happening here in prokaryotic cells is you see transcription. There's your DNA molecule. There's transcription occurring. And what you see down here is actually you see proteins being made. This is translation that's occurring down here. That's a ribosome, and these are proteins that are being made. Okay? We see that even while transcription is occurring in prokaryotes, translation is also occurring. And that's because they're both in the same place at the same time. That's impossible to happen in eukaryotic cells. In eukaryotic cells, we have a nucleus. And that nucleus is where the transcription occurs. And after transcription occurs, there's a significant amount of processing that occurs that I'll talk about. And then, if this is a messenger RNA, it's moved out. And what happens? We see translation occurring. Translation is occurring in the cytoplasm in eukaryotes, but transcription is occurring in the nucleus. They're physically separated. Okay. These have some pretty big implications, actually, for control of expression. We'll talk a little bit about those again when I talk about gene expression. Eukaryotes, as I noted earlier, have 3 RNA polymerases. Right? RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Okay? You can see that RNA polymerase 1 primarily makes the ribosomal RNAs. It at least makes all the big ribosomal RNAs. RNA polymerase II makes the messenger RNAs. It also makes some of the small nuclear RNAs that I'll talk about in a bit. RNA polymerase III makes the tRNAs and the 5S ribosomal RNAs. These are the smallest RNAs except for these guys over here. Right? So these are relatively small RNAs, tRNAs and 5S ribosomal RNA. Now these are specialized. And these have specialized promoters that are specific for the systems of transcribing each one of these. And we'll see there are, in fact, systems that are involved in doing that. What you see on the screen is an extraordinarily potent poison. It kills a fair number of people every year. It's called alpha-amanitin, and it's made by certain mushrooms known as death caps. Okay? Death caps. Alpha-amanitin is exquisitely poisonous because what it does is it inhibits all of the RNA polymerases in a eukaryote. And most importantly, RNA polymerase II, which makes the messenger RNAs, is very sensitive to it. A little bit of this and you're in trouble. Okay? Every year there are hundreds of, I shouldn't say hundreds, but there are dozens of people who die from having eaten what they thought were safe mushrooms. Okay? And they've gotten a hold of some death caps. If you ingest death cap mushrooms, okay, you have a matter of a few hours in which to get a liver transplant. That's your only hope. Your only hope. You've got a few hours. Okay? Now, you know how long transplant lists are. You know, as medical professionals, what's involved in doing something like that. That's your window to survive. Be careful, the mushrooms that you eat. All right. So alpha-amanitin is actually used in the laboratory as a way of studying transcription, and partly because it is so very effective at what it does. There's the source, and I don't know mushrooms well enough that I could tell that from another one myself, so I worry about that. Okay. Well, when we look at the transcription in eukaryotes, I said that we had different systems. We have a system for an RNA polymerase I, we have a system for an RNA polymerase II, and we have a system for an RNA polymerase III. And no, I'm not going to make you memorize this figure, but I do want to point out some general features of it to you. All right? We're going to focus most of our attention right here on RNA polymerase II. Okay? And RNA polymerase II, what we discover is that there are what we call control elements. When you hear the word element, we're not talking about boron or silicon or something like that. In eukaryotic cells, we're talking about specific sequences. And these specific sequences play roles in regulating transcription. The situation in eukaryotic cells, as I said, is much more complicated than prokaryotic cells. A promoter pretty much takes care of things in a prokaryotic cell. In a eukaryotic cell, we have to have multiple elements there. Why does it have to be so complex? Well, because we are so complex. We only have about 10 times as many genes as E. coli does. Only about 10 times as many genes as E. coli does. But think of the different types of cells that we have in our body. We have bone cells, we have muscle cells, we have skin cells, we have pancreas cells. You name it, we have different specialized cells. And the way that those cells are different from each other is a function of the proteins that are in them. We don't want my bone cells to be making things that my muscle cells would be using, for example. Maybe some we want, others we don't. So the mixing and matching, the pattern of proteins that I want in any given cell is going to be very complicated. It's not that for E. coli. E. coli has to do the following. Is sugar here? Okay, let's break it down and divide. Right? That's basically what E. coli has to decide. Right? For a eukaryotic cell, am I a bone cell? Am I a bonehead? What type of cell am I? Right? These are very important things, and so it takes a very complicated system to control that. We're going to scratch the surface of that. Do the same things exist for RNA polymerase 1 and for RNA polymerase 3? Well, not quite as complicated. But we discover that, nonetheless, they have different promoter, what they're called elements, in this case promoter elements, that help to regulate how much of these given genes are made. That sort of makes sense if you think about it. You say, well, don't we have a constant need for ribosomal RNA or a constant need for transfer RNA? And it kind of depends on the cells. Some of the cells in our body are very quiescent. They don't do much. They don't have as many needs. No reason to waste energy on making things that you're not going to need. Other cells in our body are dividing very rapidly. Our intestinal cells that I've talked about will do that. Okay? So we have to think about that in terms of the bigger picture. All right, let's see. Eukaryotic cells also have TATAAT boxes. The TATAAT box wasn't something that was unique to E. coli. The TATAAT box in eukaryotic cells is somewhat different than E. coli. The sequence is very similar, TATAAA, all right, which is where the TATA actually comes from. All right. This shows the percentage of the time each one of these appears at this region. Now, the different thing about eukaryotic cells is the TATA box is not set at one specific place. In E. coli, it's about -10. In eukaryotic cells, it can be anywhere from about -35 up to about -100. It'd be quite a ways away. Nonetheless, there's a TATA box. It's not an absolute thing in eukaryotic cells. Most prokaryotic cells have a TATA box. You saw some examples where they didn't, but most prokaryotic cells, their genes, have TATA boxes. All right. Genes that are made in high quantities in eukaryotic cells will, in fact, have a TATA box. Okay. So a TATA box is there, and TATA box plays an important role in getting the transcription process started. Now, that's probably a good place to do this. Let's sing. Okay? That kind of relieved everything, didn't it? You guys want to join me? Phosphodiesters are the bonds of RNA that support a ribopolymer made of G, C, U, and A. The RNA polymerase binds to a TATA box and copies from the template strand all along the way it walks. Initiation of transcription thus proceeds. From the closed to open complexing in the DNA it reads. The sigma factor gets released, its work is over fast. Polymerase can then advance after this step has been passed. In elongation, the polymerizing spree moves along the way and fits and starts synthesizing 523. The RNA is floppy and it dangles from one end. Oh, that's a most important thing for you to comprehend. Then termination finishes the RNAs, thanks to protein row or hair point forms that release polymerase. So this describes transcription steps in three-part harmonies. Here's hoping that this melody, you can learn it all with ease. Odd place to stop, isn't it? Okay.